In this lecture, I'll introduce some of the key notions that we'll be using in this module. Things like bioethics, biotechnology, some techniques that we will be covering throughout the semester. There are other ways to define these key notions, and it will be instructive for you to look up these definitions and compare them to the ones that I'll introduce in what follows. So first, I'll discuss the definitions of bioethics and biotechnology that we will be using, and then illustrate them with the help of a couple of examples. For the purpose of this module, we'll define bioethics as the discipline that studies the actions permitted by biotechnology, actions like cloning or genetic engineering, among others, and asks whether or not these actions are morally acceptable, and if so, how we should manage them socially in order to promote citizens' welfare, protect their rights, and treat them fairly. So bioethics covers both moral and social questions that arise from the actions made possible by different types of biotechnologies. So bioethics tries to settle whether actions made possible by novel technologies are morally permissible or not. So in order to understand the nature of bioethics, we need to unpack the notion of biotechnology as well. The definition we're relying on goes as follows. Biotechnology is the application of science and technology to living organisms and their parts, or to products and models of living organisms, in the hope of producing understanding, goods, and services. So biotechnology concerns the application of science and technology to either models or actual biological organisms and their parts. The aim of biotechnology is to generate understanding of certain aspects of these biological organisms, how they work, to produce certain goods and services. There are two more general observations about the field of biotechnology that are worth highlighting at this stage. Namely, biotechnology is a multidisciplinary field. This means that it involves many different disciplines, both pure and applied, things like genomics, molecular biology, cell biology, biochemistry, chemical engineering, data science and computer science, and so on. It is also interdisciplinary, which means that these disciplines work together to achieve the goals of biotechnology. Another important fact that's worth stressing is that the term bio is meant to reflect the fact that the efforts carried out in these disciplines apply to biological organisms, and not the fact that all of the disciplines involved are biological disciplines. Rather, biotechnology can be seen as the application of science and technology to biology. Perhaps a better feel for what biotechnology is all about might come from considering some basic examples of work carried out in biotechnology. Take, for instance, physicists and engineers who work together to produce nanovehicles. These are vehicles small enough to enter the bloodstream and deliver drugs to cure various diseases. Or, another example, geneticists and information technologists work together for finding strategies to eliminate adverse drug effects and make precision and personalized medicine a reality. Finally, engineers in biotechnologies can work together to engineer organisms that will alert us or even remove pollutants from the environment. Now I want us to look at how the actions made possible by biotechnology can give rise to ethical and social issues or questions. Since biotechnologies enable us to make many things human beings have never been able to do before, it has generated and will continue to generate many new ethical issues. Issues concerning what we should and shouldn't do, and many new social issues. Issues concerning what a society should and shouldn't allow, or should or shouldn't fund. So there is a distinction we'll be talking more about between ethical issues, which concerns what is the right thing to do for individuals or for states, and social issues, which concerns what societies or governments should allow or should fund. The same biotechnology used in different contexts and for different purposes can give rise to multiple ethical and social issues. So there is not a one-to-one straightforward mapping between biotechnology and some bioethical issue. In what follows, I will guide you through a very short list of such possible mappings from specific biotechnologies to ethical and social issues, just to give you an overview of the sort of things we will be discussing throughout the semester. Don't worry too much about losing track of what maps into what at this stage. So depending on the context in which a biotechnology is used, it can give rise to different ethical issues and different social issues. So there is not a one-to-one mapping between the two. I will start with genetic engineering. This covers techniques which involve the introduction, elimination, and modification of genes so as to affect the properties or behaviors of an individual human, plant, or non-human animal. Ethical issues associated with genetic engineering include designer babies, that is, babies genetically modified for beauty, intelligence, or to be free of disease, genetically modified foods and the challenges they pose, both ethically and socially, the human use of animals, and other environmental issues arising from the widespread of both genetically modified foods and the human's use of animals. As I already mentioned the difficult ethical issues raised by the possibility of designer babies, we could extend that list by thinking about techniques of assisted reproduction. Under this umbrella term, one finds all the techniques via which individuals or couples having trouble conceiving can be helped to have a healthy baby. Things like intrauterine insemination, in vitro fertilization, assisted hatching, and so on. Some of the ethical and social challenges that arise from the use of these technologies concern reproductive freedom, screening and embryo selection, as well as ethical and social issues concerning the resources of reproduction. Cloning, the process of generating a genetically identical copy of a cell or an organism, happens often in nature. For example, when a cell replicates itself asexually without any genetic alteration or recombination. In biomedical research, cloning is broadly defined to mean duplication of any kind of biological material for scientific study, such as a piece of DNA or an individual cell. The type of cloning that is the focus of much ethical controversy involves the generation of cloned embryos, particularly those of humans, which are genetically identical to the organisms from which they are derived, and the subsequent use of these embryos for research, therapeutic, or reproductive processes. When discussing cloning, we need to distinguish between reproductive and therapeutic cloning. Very briefly, reproductive cloning involves the implantation of a cloned embryo into a real or artificial uterus. The embryo develops into a fetus that is then carried to term. Reproductive cloning techniques underwent significant change in the 1990s following the birth of Dolly, the first cloned non-human animal. Therapeutic cloning, on the other hand, is intended to use cloned embryos for the purpose of extracting stem cells from them without ever implanting the embryos in a womb. Therapeutic cloning enables the cultivation of stem cells that are genetically identical to a patient. The stem cells could be stimulated to differentiate into any of the more than 200 cell types in the human body. The differentiated cells then could be transplanted into the patient to replace diseased or damaged cells without the risk of rejection by the immune system. These cells could be used to treat a variety of conditions, including Alzheimer's disease, Parkinson's disease, diabetes, stroke, and spinal cord injury. In addition, stem cells could be used for in vitro or laboratory studies of normal and abnormal embryo development or for testing drugs to see if they are toxic or cause birth defects. So therapeutic cloning uses cloned embryos to extract stem cells, which in turn can be used for research or for future therapies. The list of ethical and social issues triggered by different cloning techniques is long, but a representative sample would include ethical concerns about human dignity and individuality in the case of hypothetical cloning of human embryos, issues concerning the control of birth defects in cloned individuals in cases of both human and non-human animal cloning, as well as broader animal ethical issues and social and ethical issues concerning the therapeutic applications of cloning techniques. Since I already mentioned therapeutic applications of biotechnological products, I will also say something about a newer branch of biotechnology. Pharmacogenomics, or pharmacogenetics, refers to the study of genetic variations that determine an individual's metabolism and response to various drugs. So, this relatively new field of pharmacogenetics, which combines pharmacology, the science of drugs, and genomics, the study of genes and their functions, aims to develop effective, safe medication and doses that will be tailored to a person's genetic makeup. Many drugs that are currently available are one size fits all, but they don't work the same way for everyone. It can be difficult to predict who will benefit from a medication, who will not respond at all, and who will experience negative side effects, also called adverse drug reactions. So, the general aim of pharmacogenomics is to contribute to the development of personalized and more precise treatments of individuals. Even considered in these general terms, one can point to some ethical and social issues raised by the use of pharmacogenomic technologies. Questions like the following stand out. Who owns the patient data required for pharmacogenomics research? What are the standards for checking drug safety? So the ethical and social issues triggered by the introduction of pharmacogenomics concerns ownership of biodata, drug safety, the type of tests that need to be run in order to secure the success of personalized and precise medicine. During the semester, we'll be covering some of the ethical debates surrounding the biotechnologies already mentioned. We'll also be looking at technologies of life support and their implications, while some of the case studies I will assign in the seminars will cover ethical problems raised by genetic screening, nanotechnologies, and bioinformatics. In this talk, I introduced two broad definitions, that of bioethics and biotechnology. Bioethics is understood as the study of ethical and social issues which arise from biotechnology. And biotechnology as the application of science and technology to biological organisms. I listed a couple of disciplines that make up biotechnology with an eye to the general ethical and social issues to which they give rise. One general message is that there is no one-to-one mapping between a particular biotechnology and particular ethical or social issue. Rather, as we will see throughout the semester, the application of a specific biotechnology in a specific context will give rise to more than one ethical question and as well to social concerns about what should be funded and what should be allowed in terms both of research and our application to that research to the generation of products, services, and knowledge about biological organisms.